You know, usually when we talk about treating cancer, there's this underlying expectation of finality. Like, it's a lot like weeding a garden.
Yeah, right, exactly.
You see a weed, you grab it by the stem, you pull it out by the root, and suddenly your lawn is green again. Problem solved.
Yeah, it seems pretty straightforward in theory. I mean, you apply a therapeutic treatment, the visible tumor shrinks, and the patient goes into remission.
But when you step into the world of blood cancers, specifically acute myeloid leukemia, or AML, suddenly you realize your weed whacker is just, well, completely missing the most important part.
It really is.
We're looking at a therapeutic landscape that is honestly incredibly frustrating.
Oh, it really is the definition of a clinical uphill battle.
Yeah.
Because in leukemia, standard chemotherapy acts exactly like a lawnmower.
Right.
It aggressively chops off the visible top of the weed. And in blood cancer terms, we call those the blast cells.
The blast cells, okay.
Yeah, they are the rapidly dividing cells that flood the bloodstream. So the chemo clears them out, the patient appears to go into remission, but the mower blades completely miss the roots lying perfectly intact underground.
Wow. And those roots, those are what we call leukemic stem cells, right? Or
yeah,
LSCs.
Exactly.
And if you don't kill the roots, well, the weed always grows back. The cancer inevitably returns. So welcome to another deep dive.
Thanks for having me.
Today, our mission for you is to unpack a really groundbreaking 2026 paper from the journal Cell Stem Cell. These researchers have discovered exactly how these leukemic roots manage to survive highly toxic chemotherapy environments.
Yeah, it's fascinating.
They basically build your own secret, highly specialized metabolic bunker.
And finding a way to destroy these resistant LSC roots while leaving normal, healthy blood stem cells completely unharmed, well, that is essentially the holy grail of leukemia research.
Holy grail, yeah.
Today, we are going to explore how researchers achieve this across multiple models, uncovering a hidden survival mechanism that actually challenges a lot of assumptions we have about tumor metabolism and even popular diets.
So, to start figuring out how these roots are surviving, the researchers began with a biochemical mystery.
Right.
They were looking at aggressive models of AML, and they decided to perform a metabolomics analysis.
Which is basically a massive audit.
Exactly. They took normal, healthy bone marrow, and they took the leukemic roots, and they just audited every single chemical and metabolite inside them to see what was different.
And the results are completely wild.
Right. The leukemia cells were absolutely flooded with a specific molecule called β-hydroxybutyrate, or BHB, along with its precursors like acetoacetate and HMG-CoA.
And you know, the sheer scale of this discovery is what caught everyone's attention, because this wasn't just some weird quirk happening in a plastic petri dish in a lab.
Right.
When the researchers looked at blood samples from actual human AML patients, they saw markedly higher levels of BHB compared to healthy donors.
Which is crazy on its own.
It is. But it gets stranger. When they looked at the mouse leukemia models, the plasma BHB levels in the blood were so astronomically high, the biochemical profile looked identical to subjects that were eating a strict ketogenic diet.
Like a 90% fat keto diet.
Exactly.
But they weren't on a keto diet. They were eating normal, standard, carbohydrate-heavy food.
Yeah, normal lab chow.
So let me make sure I'm tracking the biology here for everyone listening. Normally, BHB is a ketone body. It's an emergency backup fuel, right?
Right, exactly.
If you or I stop eating for a few days, or if we go on a strict keto diet, our liver realizes the body is running out of readily available sugar.
Yes.
So the liver starts breaking down body fat and turning it into these ketones, like BHB, which then travel through the blood to keep our brain from shutting down.
That is the standard, healthy physiological process, yeah. The liver is essentially the body's ketone factory.
But the liver isn't doing that here. Are you saying these rogue blood cancer cells have essentially built their own internal micro-liver, like to manufacture their own emergency rations?
That is exactly what they're doing.
Wow.
The leukemic roots are engaging in a process called autonomous ketogenesis. So instead of waiting for the liver to deliver fuel, the cancer cells are actively pulling in fats themselves.
Just slurping them up.
Yeah, and breaking them down through a furnace-like process called fatty acid oxidation, or FAO, and converting them into their own private, massive supply of ketones.
That is deeply unsettling, that a blood cell can just hijack a liver function like that.
Yeah, it really is.
But I imagine the researchers didn't just guess this was happening. How did they actually prove that cancer cells were the ones making it?
Well, they used some incredibly precise biochemical detective work. First, they used a specific drug called etomoxer, or ETO.
Okay, ETO.
An ETO acts like a bouncer at the door of the cellular furnace. It blocks the gatekeeper enzyme called CPT1A, which is what allows fatty acids to even enter the burning process.
Ah, I see.
The moment they introduce this drug and block the fat from getting in, the BHB levels in the cancer cells plummeted.
Okay, so cut off the raw material, the factory stops.
Right. But to be absolutely undeniably certain, they essentially put a microscopic GPS tracker on a fat molecule.
A GPS tracker on fat. That's amazing.
Yeah, they tagged palmitate, a type of fat, with a heavy carbon-13 isotope. They fed it to the cells and then literally watched as the cancer cells dismantled that specific tagged fat and reassembled it into heavy carbon BHB ketones.
They literally watched it happen.
They did. And when they used genetic engineering to completely delete the CPT1A gene, the one responsible for that gatekeeper enzyme, the factory was permanently shut down.
Wow.
The roots lost their ketones entirely.
It's an airtight case. So let's take a quick second to recap this first major mechanism for you listening at home.
Good idea.
We have these leukemic stem cells, the LSCs, acting as the indestructible roots of the cancer.
Right.
They're autonomously pulling in fat and producing their own ketone fuel, BHB, via fatty acid oxidation.
Yeah.
And they're doing this at such a massive scale that their internal environment mimics a full-blown systemic keto diet.
Exactly.
Which, immediately begs a question for you to ponder. If we know these cancer roots have built this rogue fuel factory, what happens if we find the main power switch and turn it off completely?
Well, that is the exact logical leap the researchers took.
Right.
Because if you want to shut down a complex manufacturing process, you don't go after the individual assembly line workers.
No, you go after the boss.
Or the main bottleneck. You find the rate-limiting enzyme.
And a rate-limiting enzyme, just to clarify for everyone, is the slowest or most critical step in a chemical chain reaction.
Exactly.
If you control that one step, you control the entire output of the factory.
Precisely. And they found it. The key suspect is an enzyme called HMGCS2.
HMGCS2. Okay.
And what made the researchers realize they had struck gold was the location of this enzyme.
Where was it?
HMGCS2 was highly expressed, meaning very active, inside the leukemic roots. But it was barely present in normal, healthy bone marrow. And it wasn't even really active in the bulk leukemic blast cells, the top of the weed. It was highly specific to just the roots.
So they have a specific target, and the next step in biology is always to see what happens when you break the target, right? So they went in and genetically deleted the HMGCS2 gene across multiple highly aggressive leukemia models like MLA-AF9 and HOXA9-1. What actually happened to the cancer?
The outcomes were incredibly dramatic. The moment HMGCS2 was removed, the ketone levels flatlined.
Makes sense.
But more importantly, the leukemic roots lost their stemness.
Stemness. Let's unpack that, because that's a crucial concept here. What makes a stem cell in this context?
It's their ability to hide and rebuild. Leukemic stem cells often exist in a state of quiescence.
Quiescence, meaning they're asleep.
Basically, think of it like a bear hibernating in a cave. When a patient gets chemotherapy, the chemo acts like a raging blizzard outside.
Okay, I like this analogy.
It kills all the active dividing cancer cells out in the open. But the LSCs, they are asleep in their cave.
The chemo doesn't even see them.
Right. The chemo doesn't recognize them, so they survive the storm. And when the blizzard passes, they wake up, multiply, and the cancer relapses.
So taking away this HMGCS2 enzyme does what to the bear?
It essentially kicks the bear out of the cage.
Oh, wow.
Without that enzyme, the leukemic roots lost their quiet, resting state. They woke up, they stopped being able to self-renew, they accumulated massive amounts of internal cellular stress, and they died.
That's incredible.
Yeah, the blast cells vanished from the bloodstream, and the subjects lived significantly longer. The researchers even took cells from those cured models and transplanted them into entirely new subjects, secondary transplantations, to see if the cancer could somehow regenerate.
And could it?
It couldn't. The roots were truly, definitively dead.
Now, I have to stop you there, because whenever I hear a phrase like, we found a way to completely destroy the cancer cells, my immediate next thought as a skeptic is always, okay, but what else did we destroy in the process?
Sure, the toxicity question.
Right. We see this constantly in oncology. A new drug obliterates the tumor, but it also heavily damages the patient's immune system or their liver or their gut lining.
Yeah.
If we shut down this ketone factory, what happens to the innocent bystanders?
That is the most vital question in drug development,
Right.
and it leads to perhaps the most significant clinical finding in this entire study.
Okay.
When they deleted that exact same HMGCS2 gene in normal, healthy hematopoietic stem cells, the HSCs responsible for generating your daily blood supply, absolutely nothing happened.
Wait, nothing at all.
Completely fine. Normal blood development continued without skipping a beat.
That's amazing.
It turns out that HMGCS2 is entirely dispensable for normal blood cells. They just don't need it to survive. It is a highly specific vulnerability that only the cancer relies on.
So it's the perfect target.
Exactly. And they validated this rigorously. They used patient-derived xenog models, PDX models, where actual human leukemia cells are grown in immunodeficient mice.
Just to ensure they weren't just curing a quirk of mouse biology.
Right. And when they silenced HMGCS2 in those human leukemia models, it suppressed the disease just as effectively without harming healthy blood production.
That is the ultimate clinical holy grail.
Yeah.
Maximum toxicity to the cancer roots, zero toxicity to the healthy roots. So let's do another quick recap for you guys listening.
Let's do it.
We found the bottleneck. The enzyme HMGCS2 is the main switch for making this ketone fuel.
Yep.
Deleting it forces the leukemia roots out of hiding, strips them of their regenerative power, and kills them.
Right.
And it does all of this while completely sparing the normal blood cells.
Exactly.
Which is fantastic, but it raises a massive mechanistic question for us to figure out next. We broke the ketone factory and the cells died. But how exactly did they die?
That's the real mystery.
Right. What was the actual murder weapon once that ketone shield was lowered?
Well, to understand the murder weapon, we have to talk about a very specific, almost violent type of cell death, kind of ferroptosis.
Ferroptosis, which is very different from how cells normally die, right?
Very different.
Because usually if a cell gets too damaged or old, it undergoes apoptosis, which is this very neat, orderly, programmed cellular suicide. The cell politely packs up its machinery, shrinks down, and allows the immune system to clear it away.
Exactly. Apoptosis is orderly. Ferroptosis is chaos.
Chaos.
Yeah, it is a form of cell death driven by iron and intense oxidative stress, and its primary target is the outer boundary of the cell itself, the lipid membrane.
Okay, so if I picture the cell membrane as a protective metal barrier around a city, ferroptosis isn't someone dismantling the barrier slowly.
No.
It's like the metal rapidly rusting from the inside out.
That's a highly accurate visual.
The structural integrity just degrades and crumbles until the whole thing falls apart.
That is exactly what happens. When the researchers looked at the dying leukemic roots after removing the HMGCS2 enzyme, they saw exactly this rusting process.
What did it look like?
They found massive levels of reactive oxygen species, or ROS, swirling around inside the cells.
Just causing damage.
Yeah. They even looked under an electron microscope and saw that the mitochondria, the power plants of the cell, were physically shattered. Their internal folds, the cristae, were broken and fragmented.
Wow.
But the real smoking gun was found in the lipids.
The fats making up the cell membrane.
Right. They ran an audit of all the fats in the cell membrane, a lipidomics analysis, and they found a massive abnormal spike in a specific type of fat called polyunsaturated fatty acid phospholipids, or PUFA-PLs.
Okay, PUFA-PLs. Why are PUFAs so dangerous to the cell?
It comes down to their chemical structure. Polyunsaturated fats have multiple double bonds in their carbon chains.
Okay.
And in biochemistry, double bonds are highly reactive. They are incredibly susceptible to oxidation.
So they rust easily.
Exactly. If we use your metal barrier analogy, PUFAs are like building the barrier out of highly reactive, ungalvanized iron.
Oh, that's bad.
Or to use another analogy, if the inside of the cell is a room, PUFAs are dry kindling scattered all over the floor.
Okay, I see where this is going.
When that HMGCS2 enzyme was removed, this dry kindling piled up uncontrollably inside the cancer cells. The reactive oxygen species acted as the spark, and the result was a massive ferroptosis fire that burned the cell down.
So if removing the ketones causes the kindling to pile up and burn, that means the BHB ketones were originally acting as a flame retardant.
Precisely. They were an anti-rust coating.
That's wild. But how did they prove that connection?
The researchers proved this causality with two brilliant rescue experiments.
Okay, let's hear them.
First, they took the dying cancer cells that had their HMGCS2 gene deleted, and they treated them with a specific pharmaceutical drug called leproxtatin-1.
Liproxtatin 1, what does that do?
It is designed purely to block ferroptosis. It essentially acts as a chemical fire blanket.
A fire blanket, okay.
When they applied this drug, the cancer cells miraculously survived the genetic deletion.
Wow, meaning the only reason they were dying was a rusting process.
Exactly.
And what was the second rescue mission?
They simply bypassed the broken factory. They manually gave the mice BHB supplements.
Wait, they just fed them the ketones?
Yeah, they just handed the cancer cells the exact ketone fuel they could no longer manufacture themselves. And when they did that, the leukemia came roaring back to life.
Oh my goodness.
This proved definitively that the entire evolutionary purpose of building that internal microliver was to create a lipid shield preventing ferroptosis.
That is just mind-blowing biology. Okay, time for another quick recap for you listening. Let's make sure we have all the pieces.
Let's do it.
Without their BHB fuel, these leukemic stem cells start stockpiling highly flammable lipids, the PUFA-PLs, in their membranes.
Yes.
The combination of those flammable lipids and internal cellular stress triggers ferroptosis. It literally rusts the cell to death.
Right.
But if you block ferroptosis with a drug, or if you artificially supply the BHB back, you save the cancer.
You do.
Which leaves us with an even deeper puzzle to solve. Because, BHB is just a piece of metabolic fuel, right? It's just energy.
Right.
How does a simple fuel molecule like BHB tell a complex cell to stop stockpiling flammable lipids? How does fuel turn into a genetic instruction?
To answer that, we have to dive into the final and arguably most fascinating mechanism in the paper, epigenetics.
Epigenetics. I love this.
Because it turns out BHB isn't just being thrown into the cellular furnace to burn for energy. It is actually moonlighting as a chemical messenger.
I love epigenetics because it fundamentally changes how we think about DNA.
Yeah.
People often think of DNA as a rigid blueprint, but it's really not.
Not at all. Your DNA is incredibly long. I mean, if you stretched it out, it would be about 2 meters long, but it has to fit inside a microscopic nucleus.
Right.
To do that, the cell wraps the DNA tightly around tiny protein spools called histones.
So if the DNA is a massive multi-volume instruction manual, the histones are the spools keeping certain chapters closed.
Yes, exactly.
As the DNA is wrapped tightly around a spool, that specific chapter of the manual is closed and the cell can't read the instructions.
Yeah.
But if the spool unwinds, the chapter opens, and the cell executes those specific instructions.
Exactly. Epigenetics is the study of how those spools are controlled. And what the researchers discovered is that the BHB ketone travels straight from the metabolic factory into the nucleus of the cell.
Yeah.
Once there, it physically attaches itself to those histone spools.
So the fuel molecule is literally acting like a physical padlock on specific chapters of the instruction manual.
Yes, it's a chemical tag. This physical attachment process even has a specific name: lysine beta-hydroxybutyrylation, or KBHB for short.
KBHB. Got it. So how did they figure out which chapter was being padlocked?
To figure out exactly which chapter of the DNA manual BHB was locking up, the researchers used an advanced sequencing technique called ATAC-seq.
ATAC-seq, okay.
Yeah, it basically maps the physical accessibility of the DNA. They looked at exactly which parts of the manual opened up when BHB was removed.
And what chapter were the ketones keeping locked?
A single specific gene called FADS2.
FADS2. What does FADS2 do?
FADS2 is the code for a fatty acid desaturase enzyme. In plain English, FADS2 is the exact cellular machinery responsible for manufacturing those highly flammable POFA lipids we talked about earlier.
Oh. Okay, let me see if I can string this entire incredible Rube Goldberg machine together from top to bottom.
Go for it.
In a normal, healthy leukemic root, which is a terrible phrase, but you know what I mean.
Yeah, the thriving cancer cell.
Right. The cancer cell autonomously burns fat to make massive amounts of BHB ketones.
Oh.
That BHB travels into the nucleus and acts as a padlock on the FADS2 gene.
Exactly.
Because the FADS2 chapter is locked, the cell cannot manufacture the FADS2 enzyme.
Right.
Because that enzyme is missing, the flammable POFA lipids stay extremely low.
Yep.
And because the flammable lipids are low, the cell's membrane is completely stable, making it immune to rusting to death via ferroptosis. It survives the chemotherapy storm.
That is a perfect summary of the survival mechanism.
Thank you.
And conversely, when the researchers step in and therapeutically block that initial bottleneck, HMGCS2, the entire chain reaction collapses.
Right, the factory shuts down.
The BHB levels plummet. Because there is no BHB, the padlock falls off the FADS2 gene. The chapter opens.
The cell reads it.
Exactly. The cell reads the instructions and immediately mass produces the FADS2 enzyme. That enzyme rapidly fills the cell membrane with highly flammable lipids, and ferroptosis completely destroys the cancer.
And I assume they proved this FADS2 connection directly, right?
They did. For their final definitive proof, they went into those highly vulnerable, dying leukemia cells that had their ketone factory broken. But this time, they artificially silenced the FADS2 gene directly. They forced the chapter shut themselves.
Oh, wow.
And because FADS2 couldn't be read, the dry kindling never built up in the membrane. And the leukemia cells miraculously survived and started multiplying again, even without any BHB present.
It all ultimately hinges on keeping FADS2 turned off.
It does.
It is genuinely a masterclass in uncovering cellular causality. We start with a strange metabolic observation, high ketones, and trace it all the way down to the physical folding of DNA, preventing a cell membrane from oxidizing.
It's beautiful science.
Let's do one final recap for you guys before we wrap this up.
Good idea.
BHB physically alters the DNA spools, the histones.
Right.
This modification suppresses the FADS2 gene.
Yes.
And keeping FADS2 turned off prevents the buildup of flammable lipids, saving the cancer.
Exactly.
So, as we zoom out here, why should you, the listener, care about all these dense epigenetic mechanisms?
Well, it has huge real-world implications.
Right. We constantly hear in popular health culture about things like intermittent fasting or ketogenic diets. They are often pitched as universally good for you.
Or even promoted as anti-cancer lifestyles.
Exactly. The logic is usually quite simple. Cancer loves sugar, so if you cut out carbohydrates and lower your blood sugar, you starve the tumor.
And we should be clear, for certain solid tumors that are highly dependent on glucose, that metabolic logic can hold true. A ketogenic state can create an unfavorable environment for some cancers.
Context in biology is absolutely everything.
It is. This paper throws a massive evidence-based wrench into the one-size-fits-all approach to diet and disease. We just learned definitively that leukemic stem cells actively thrive on ketone bodies.
Right. They use them to dodge death.
Exactly.
If you were to go on a strict keto diet or engage in prolonged fasting while fighting acute myeloid leukemia, you might theoretically be handing the indestructible roots of your cancer the exact metabolic armor they need to survive your treatment.
It's a scary thought.
You would be pumping your blood full of free BHB, locking their FADS2 genes for them, and preventing the chemotherapy from rusting them to death.
It perfectly highlights why the future of oncology has to be personalized, targeted medicine. We can no longer just guess at a patient's metabolism or apply broad dietary rules. We have to map the specific metabolic vulnerabilities of the exact disease we are fighting.
Which leaves us with a really profound final thought for you to mull over.
Yeah, think about this. If blood cancer cells are smart enough, evolutionarily speaking, of course, to hijack our body's ancient starvation defense mechanism.
To take something like ketogenesis and use it to build a literal epigenetic shield around their DNA.
What other completely healthy metabolic states are tumors secretly exploiting right under our noses?
It really makes you wonder how many other survival mechanisms are hiding in plain sight, just waiting for the right biochemical audit to uncover them.
Exactly.
It really makes you look at that weed whacker entirely differently. We always thought we just needed a sharper blade, a more toxic chemical to kill the weed.
But maybe we just need to figure out how the weed is drawing water from the soil and turn off the tap.
Turn off the tap. I love that. Thank you for joining us on this deep dive. Keep questioning the consensus, stay curious about the world around you, and we'll see you next time.